So in our last video we were talking about food production, and obviously the key to that is plant growth and photosynthesis. So we're going to start to have a look at photosynthesis, and to do that the first thing that we need to do is have a look at light, because that is on what photosynthesis is dependent. Now when we look at white light, that is visible light that hits a leaf, then that light has different fates. What can happen is the light can either be absorbed by the leaf, or it can be reflected off the leaf, or it can be transmitted through the leaf. And we're going to have a look at some of the features of light and have a look at how light is absorbed by a leaf. And to do that, we're just going to talk about what light is. Now, light is a form of electromagnetic radiation. It travels in waves, and here you'll see that I've got a diagram of a wave here, and between this crest and this crest is what is known as a wavelength. So it is measured in something called nanometers. Now, different lights have different wavelengths. When we put a beam of white light through a prism, something that is called a spectrum is produced. So by a prism, what we mean is like this kind of plastic or glass triangular item, and it splits the light up into the different colours. So you see your kind of rainbow colours. I said a moment ago that different colours of lights have got different wavelengths. So here what we're talking about down here where we've got blue light, what we're saying is that the wavelength of that here, the distance between these crests, is shorter than, say, red light, which has got a wider wavelength. So it would be a wavelength that would be wider like that. So that's what we mean when we're talking about wavelength. And here we've got different coloured lights at different wavelengths. So you've got your red, your orange, your yellows, your greens, your blues and your violets. So when we're looking at light being absorbed by a leaf, you have to think of all these different coloured wavelengths hitting that leaf, and we'll look at the implications of that just shortly. Now, the leaves themselves have got certain coloured pigments. Chlorophyll is the most important one, but we'll have a look at some of the other pigments that are there as well. Now, these pigments that are there absorb the light energy, and the pigments absorb different wavelengths of light. Now, we're going to just have a look at what we mean by a pigment first of all. So to do this, we're going to have a look at our glass of red wine that is here. Now, the red wine that's here has got a red pigment. Now, when we use the word pigment, what we're saying is a pigment is a substance that absorbs visible light. Now, in this situation that's here, we've got white light hitting our glass of red wine. And when that light hits the glass of red wine, we've got our blue there, we've got our green, we've got our indigo, etc., violet. And that all gets absorbed in this pigment here. So it's absorbed by this red wine here. But what is not absorbed is the actual red. Red is reflected or bounces back, or the red is transmitted through the red wine, and that is why a glass of red wine appears red. Now, if you think of this in terms of if we were to change our glass of red wine here to a glass of green chlorophyll, when white light hits a glass of green chlorophyll, then what happens is all the colours, like our blue and our red, get absorbed in this pigment, but green is either passes through or it's reflected back. So it's not absorbed. And it's important to get that concept into your head. When you're looking at a colour, what you're actually looking at is that the reflection of that visible light bouncing back at you. So if you're looking at something that is green, it is green because the colour hasn't been absorbed into the pigment there. What is happening is the colour is being reflected back to you, or it's transmitted through the item. So what we're saying there is that a pigment displays the colour of the light that it does not absorb. And it's important to keep that key concept in your head. So the pigment displays the colour of the light that it does not absorb. Strikes a leaf, then particular pigments will absorb particular wavelengths of light. Now remember, we talked before, a moment before, just talking about pigments, that if, say, you've got the colour green, then that's meaning that that is not absorbed. So if you're thinking of your chlorophyll as being a green colour, then you would be thinking about the fact that the green wavelength isn't absorbed and it's reflected or it's transmitted through the leaf and making the leaf look green. So if we move on and have a wee look at what happens when a beam of white light is passed through a sample of these pigments, we can do that by looking at the diagrams that we've got here. Now the one that I've got down at the bottom here is just where we've got a projector down here, which is a source of white light, and the light shines through, it's put through a wee slit in this cardboard here, through a lens, and then through this prism. So you get the different wavelengths that are here. So you get this here. We talked about this earlier in the diagram. This is looking at this spectrum, the visible spectrum of light, which is here, the blue side here, the red side here, and the other colours in between. Now when we have a look at the diagram up at the top here, and what we're going to think about here is that in this part here we've got our chlorophyll pigments. So sorry, we've got our pigments, so we've got our chlorophyll and our other pigments that are there. So when our light is passed through these leaf pigments, these extracted leaf pigments, and put through the glass prism, we get what is known as an absorption spectrum. And what you get is you get these colours, there's some of these colours that are here, but you've got these black bands. Now these black bands that are here, they are showing a region in the spectrum where the light of a particular wavelength has been absorbed by the leaf pigments, and it's failed to pass through the prism and then onto the screen. So what we're seeing here is in this area here, for example, in this kind of blue region, or this kind of bluey region that is here on the spectrum down below, that has been absorbed up here. As the light is shining, it's been absorbed into these pigments here. So therefore those particular wavelengths don't show up on the board when we shine this light through. And in the same way as well, you've got areas in the red region as well, or kind of reddy orange region here as well, where there's blanks, because again, that particular wavelength has been absorbed by the leaf pigments. Now you've also got these coloured bands, and I'm going to try and see if I can just maybe take that away a wee bit. No, I can't remove these colours that are there, but you've got these coloured bands that are there. Now the coloured band, so for example, this green coloured band that is here, that is a region where a light of a particular wavelength has not been absorbed and by the pigment extract. So the chlorophyll appears green to the eye because it doesn't absorb the green light, but it reflects and transmits it, as we've talked before. We've also, there's regions here, you'll see this blue and these yellow and red, some of the red that is here as well. These are showing up on there, and that's areas as well where the light of that wavelength hasn't been absorbed by the pigment extract. So you've got in this extract that is up here as well, then as the light is passing through, if we have a wee look at this blue part here, that is not being absorbed here in this chlorophyll. What is happening is it's passing right the way through and it's showing up on the screen here. So what you need to think about when you're looking at this absorption spectrum is you need to have in your mind, well, what is being absorbed by the chlorophyll? And if it's been absorbed, or I'm saying chlorophyll, by the pigments, because there's more than just chlorophyll there, what has been absorbed? And the wavelength of light that's been absorbed does not show on the spectrum. Where there's a coloured band, that's where the light has not been absorbed by the pigment extract. So hopefully you've kind of got that picture in mind, and I want to move on to have a look at these graphs that are here. The degree at which each of the wavelengths of light are absorbed can be measured using something called a spectrometer. Now, a spectrometer is just a machine that measures the amount of absorption of the light by each of the different pigments. And if we have a look up at the top here, this is the absorption spectrum, so measuring the amount of absorption that's taking place. So we have a wee look at the diagram that is here. We've got our chlorophyll A that is here. If we have a look at that, it's showing how much light is being absorbed by that chlorophyll. And if you look down at the bottom of the diagram, I'm not sure if it's actually showing, I'll lift it up a little bit, you'll see there's the wavelengths of light down at the base, and you'll see that in this area here, that's kind of where our green colours are and our yellow colours are. So what we're really saying is when we look up at the diagram that's here, is we're showing that a lot of light or wavelengths are being absorbed here at this end here, where there's a kind of blue wavelengths, and down here we've got absorption here that's taking place down at our orange and yellows, or it's not quite into the yellows, it's more orange and red colours. So that is the absorption spectrum of chlorophyll A, and it makes sense that you've not got a lot of absorption happening in this green area, because really what is going on there is you are seeing that green colour, because that light is not, that wavelength of light isn't being absorbed. Now you'll notice as well in this graph that is here, you've got chlorophyll B, and it's absorbing here, and this is this line here, and it is absorbing a light in the blue and the red regions as well. So your chlorophyll A and B absorb light energy mainly from the blue and red regions, but there are other pigments that are there, such as your carotene or your carotenoids, and they're absorbing Here they are here. There's some absorption happening in the blue area and then happening here from this area down here, which is maybe, if I have a wee look, if I move that up, let's just have a wee look, that's in this orange region that is down here as well. So that means that you've got a range of absorptions that are taking place. You know, if you only had chlorophyll A, for example, you would only have absorption taking place just on this area here that is on the graph. Whereas if you've got these other pigments that are there, then there could be absorption of light at different wavelengths. Now the second graph I want you to have a look at is this one here, looking at the action spectrum. Now the action spectrum is looking at the photosynthetic rate, or the rate of photosynthesis at the different wavelengths, and how much photosynthesis is taking place at those wavelengths. And that is measured in the way that you've done before when you've been measuring photosynthetic rate. So for example, you've got your kabomba inside your test tube and you count the number of bubbles of oxygen that are coming off per minute, and you can work out a photosynthetic rate by that. So in this case here, what has happened is different colours of filters have been used, and the photosynthetic rate in these different colours has been calculated. And you'll see there's a graph that is formed like this one that is here. Now if we put these two graphs together, the absorption spectrum and the action spectrum, then we get something that looks like this, where over on this side here we've got the rate of photosynthesis with this broken line, and over on this side here we've got the absorption spectrum, and in this case here it's of chlorophyll A. So you'll see that this straight line here, here's the absorption spectrum of the chlorophyll A here, and here is the rate of photosynthesis, this line that is here. So you'll notice that where you've got a lot of absorption taking place, you get a lot of photosynthesis taking place. Where there's lower levels of absorption, you get lower levels of photosynthesis. And here you've got another peak of absorption and you get an increased rate in photosynthesis. Now this has only got the one chlorophyll in it here, but remember you've got your chlorophyll A, B, your chlorophyll B, and your carotene and your xanthophyll there. So you've got other pigments that are there. Now these we refer to the pigments such as your carotene or your xanthophyll. We refer to them as accessory pigments. Now, so carotenoids allow the plant to carry out photosynthesis in a wider range of light wavelengths. They're known as accessory pigments because what they do is they pass the energy they capture onto chlorophyll A and chlorophyll B. And the advantage to the plant of having these different pigments is that they can absorb light at different wavelengths. And this is an advantage to the plant because it extends the range of wavelengths that the plant can use for photosynthesis. So that is a summary of the light and the absorption of light by your plant, and in the next video what we'll do is we'll move on to actually have a look at the process of photosynthesis. Now that we've had a look at that light being absorbed by the plant, what then happens in the rest of the stages of photosynthesis. Now some of this is quite detailed and it can be a little bit complex. There'll be an added video that I will include with this that's been put together by somebody else, and we'll also do some practicals in the class that will help to show some of these or illustrate some of these things that are in this video.